हेलो फ्रेंड्स, आज स्टार्ट करते हैं न्यू चैप्टर बॉडी फ्लूइड एंड सर्कुलेशन का पार्ट 1। और आज आरबीसी, डब्ल्यूबीसी, प्लेटलेट्स, आर फैक्टर के बारे में पढ़ेंगे। बॉडी फ्लूइड एंड सर्कुलेशन। Vascular connective tissue. There are two types of vascular connective tissue: blood, limbs. Matrix is liquid and fiber-free. Blood. A study of blood. Hematology. Process of blood formation. Hemopoiesis in bone marrow. Color red. pH 7.4, slightly alkaline. By weight 7 to 8% of body weight. By volume 5 to 6 liter in male and 4 to 5 liter in female. Blood is false connective tissue. Cells of blood have no power of division. Fibers are completely absent in blood. Matrix of blood is produced and synthesized by liver and lymphoid organs. Composition of blood. Liquid part matrix plasma 55%, solid part blood corpuscles 45%, RBC, WBC and platelets. Plasma. Matrix of blood is called plasma. It is pale yellow in color due to urobilinogen bilirubin. Composition of plasma. Water 90 to 92%, solid part 8 to 10%, in which inorganic and organic compounds are present. Organic part of plasma 7 to 9%, protein 6 to 7% maximum. Number A, albumin 4% maximum, produced and synthesized by liver. Smallest plasma protein responsible to maintain BCOP 28 to 32 mmHg. B, globulin 2 to 2.5%. Ratio of albumin and globulin is 2:1. Produced and secreted by liver and lymphoid organs. Transport to carry substances in body. Destroy bacteria, viruses and toxic substances in blood. 3 types of globulins are present. Number first, alpha globulin, produced by liver, for example ceruloplasmin, copper carrying protein. Number 2, beta globulin, produced by liver, example transferrin, iron carrying protein. Number 3, gamma globulin, produced by lymphoid organ, present in the form of antibodies which destroy bacteria, virus and toxic substance, also called immunoglobulin. These are 5 types: IgG, gamma immuno, IgA, alpha immunoglobulin, IgM, microimmuno, IgD, delta immuno, IgE immuno. Prothrombin 0.3% produced by liver. Fibrinogen 0.3% produced by liver. Largest plasma protein, help in blood clotting. Blood corpuscles. Erythrocytes, red blood corpuscles, RBC. Number first, mammalian RBCs are biconcave, circular and non-nucleated. Number second, at the time of origin, nucleus is present in the RBC, but it degenerates during maturation process. Biconcave shape of RBC increased surface area. Due to absence of nucleus and presence of biconcave shape, more hemoglobin can be felt in RBC. Exception: camel and llama are mammals with biconcave oval shape. In RBC, endoplasmic reticulum is absent, so endoskeleton is composed of structural protein, fats and cholesterol present in the form of network called stromatin, which is a spongy cytoskeleton. Plasma membrane of RBC is called Donham's membrane. It is highly permeable. Some ion like chlorine and bicarbonate ion are impermeable to sodium and potassium ions. It is called Donan's phenomenon. Due to presence of stromatin, spongy cytoskeleton, and flexible plasma membrane, RBC 7.5 micro can pass through less diameter blood capillaries 5 micro. In RBC, higher cell organelles like mitochondria and Golgi complex is absent. Due to absence of mitochondria, anaerobic respiration takes place in RBC. In RBC, enzyme of glycolysis process are present while enzyme of Krebs cycle are absent. Antigen of blood group is present on the surface of RBC. If Rh antigen is present, then it is also found on the surface of RBC. Single RBC is pale yellow in color, while the group of RBC appear in red color. In RBC, red color respiratory pigment hemoglobin is present. In each RBC, 26.5 crores molecules of hemoglobin are present. Molecular weight of each molecule of hemoglobin 67,200 Dalton. In composition of RBC, 60% H2O and 40% solid part is present. Only hemoglobin constitutes 36% of total weight of RBC and 90% on dry weight. Hemoglobin. It is composed of two components, heme, globin. Heme 5%, globin 95% protein part. Heme iron and porphyrin. Iron present in the form of Fe+2. Each molecule of hemoglobin carries 4 molecules of O2. Globin. Each molecule of globin protein is composed of 4 polypeptide chains. Polypeptide chains are of 4 types. Alpha polypeptide chain having 141 amino acids. Beta polypeptide chain having 146 amino acids. Gamma polypeptide chain having 146 amino acid and delta polypeptide chain having 146 amino acid. On the basis of this polypeptide chain, 3 types of hemoglobin are formed in human. Hemoglobin A1 adult hemoglobin 2 alpha plus 2 beta. Hemoglobin A2 adult 2 hemoglobin 2 alpha plus 2 gamma 2 delta. Hemoglobin F fetal hemoglobin 2 alpha plus 2 gamma. Oxygen binding capacity of fetal hemoglobin is more than adult hemoglobin. Size of RBC human 7.5 micro. Change in the size of RBC is called as anisocytosis. Due to vitamin B12 deficiency, RBC become larger in size called as macrocytocytes. These are immature RBC which are destroyed in spleen. In these RBCs, amount of hemoglobin is normal. Number 2, due to iron deficiency, RBC become smaller in size called as microcytes. They are also destroyed in spleen. In the RBCs, amount of hemoglobin is less. Shape of RBC. Biconcave change in the shape of RBC is called poikilocytosis. Uremia. RBC become irregular in shape. Sickle cell anemia. RBC becomes sickle-shaped. If RBC is kept in hypertonic solution, it will shrink. Certination. In hypertonic solution, it will burst. Lifespan of RBC is 120 days. Average lifespan of RBC in all mammals 120 to 107 days. RBC count. Number of RBC in per cubic mm of blood is called RBC count. Human male 5.5 million, human female 4.5 million, newly born baby 6.8 million, rabbit 7 million, frog 0.4 million. Decrease in RBC count condition is called anemia. First, macrocytic anemia. Due to vitamin B12 deficiency, macrocytes are formed which are destroyed in spleen. Second, microcytic anemia. Due to iron deficiency, microcytes are formed. Normocytic anemia. Excess blood loss. Formation of RBC. Process of formation of RBC is called erythropoiesis. Organs which produce RBC is called erythropoietic organ. Hormone which stimulate erythropoiesis is called erythropoietin, synthesized by kidney and little quantity by liver. First RBC produced by yolk sac. During embryonic life, RBC are produced by liver, spleen, placenta, thymus gland. In adult stage, RBC is produced by RBM, which filled in between trabeculæ of spongy bones. 1% RBC are destroyed daily, but in same number new RBC are entered in the blood. Destruction of RBC occur in spleen, so spleen is called graveyard of RBC. Spleen stores excess blood corpuscles, so it is called blood bank of body. WBC. Acidophils. Size 10 to 14 micro. Basophils size 8 to 10 micro, smallest granulocytes. Neutrophils 10 to 12 micro. Monocytes 12 to 20 micro. Lymphocytes 6 to 16 micro. Lifespan acidophils 14 hours, basophils 10 hours, neutrophils 12 hours, monocytes less than 24 hours in blood, lymphocytes 5 to 7 days in blood. Stain with acidic dye like basic dye like methylene blue. Any dye acidic या basic या neutral. Shape of nucleus. Acidophils bilobed, basophils 2 or 3 lobed S shape, neutrophils 3 to 5 lobed, monocyte kidney या bean shaped. Lymphocytes large, due to which cytoplasm becomes peripheral. Functions. Acidophils they protect body against allergy and parasitic infections. Basophils secrete and transport heparin, histamine and serotonin. Neutrophils phagocytic in nature, destroy bacteria and viruses by phagocyte. Monocyte also called scavengers of blood because they engulf damaged or dead and minute bits of blood corpuscles. Lymphocytes T killer direct kill microbes. T helper stimulate B lymphocyte to produce antibodies. T suppressor suppresses T killer and protect immune system. B lymphocytes produce and transport antibodies. A special point. Number 2 to 3% of total leukocyte count, 0.5 to 1% of total leukocyte count, 60 to 65%, 6 to 8% of total leukocyte count, 20 to 25% of total leukocyte count. A special point, acidophils called eosinophilia occurs during teniasis ascaris, have fever. Due to their smaller size and phagocytic nature, they are called micropolicemen of blood. Neutrophils. Macropolicemen, monocytes. Diagram. WBC. White blood corpuscles are called as leukocyte because they are colorless. Total leukocyte count. Number of WBC per mmq 6000 to 8000 per mmq. Leukocytosis increase in total leukocyte count. This condition occurs in bacterial and viral infection. Leukocytopenia decrease in total leukocyte count. Normally total leukocyte count increase in bacterial and viral infection, but in typhoid and AIDS total leukocyte count decrease. Leukemia abnormal increase in total leukocyte count more than 1 lakh. It is called as blood cancer. On the basis of nucleus and nature of cytoplasm, leukocytes are of two types. Granulocytes. In their cytoplasm, granules are present which can be stained by a specific dye. Nucleus is multi-lobed and lobes are interconnected by protoplasmic strand. Due to presence of lobed nucleus, they are called as polymorphonuclear WBC, produced in bone marrow. There are three types: acidophils, basophils, neutrophils. Number 2, agranulocytes. Cytoplasm is clear and agranular. Nucleus do not divide in lobes, so called as mononuclear WBC. Produced in bone marrow, there are two types: monocytes, lymphocytes. Platelets. Size 2 to 3 micro, lifespan 2 to 4 or 5 days, count 1.5 to 3.5 lakh per mmQ, also known as thrombocytes. They are non-nucleated and derived from megakaryocytes called of cells of bone marrow. In shape, platelets are disc-like, oval-shaped or biconvex. In their cytoplasm, basophilic granules are present, which can be stained by methylene blue. Maximum part of cytoplasm is composed of contractile protein thrombobustanin. Decrease in number of blood platelets is called thrombocytopenia. Critical count of thrombocytes is 40,000 per mmQ. If number is less than critical count, then red spot or rash appears on the skin called purpura disease. Function: repair endothelium of blood vascular system by the formation of platelet plug because they have tendency to attach on gelatinous or mucilaginous surface. Synthesize thromboplastin, which help in blood clotting. Synthesize serotonin 5-hydroxytryptamine. Blood clotting. Blood flows from cut or wound, but after some time it stops automatically. It is called clotting of blood. Bleeding time 1 to 3 minutes, clotting time 2 to 8 minutes. Sometimes clots are also formed in intact blood vessels, which are of two types: thrombus clot, a static clot which grow bigger and ultimately block the blood vessels. If this clot is formed in the coronary vessels, then called as coronary thrombosis, which can cause heart attack. If found in brain, then called as cephalic thrombus, causes paralysis. Embolus clot, moving clots which flow with blood, more harmful due to their moving nature. Coagulation of blood. Blood exhibit coagulation या clotting in response to an injury or trauma. Prevent excessive loss of blood from the body. Injury trauma. Stimulate platelets in blood tissue at site of injury. Release certain factors. Initiate series of linked enzymatic reaction cascade process involving number of factors present in plasma in an inactive state. Form an enzyme complex thrombokinase. Thrombokinase prothrombin to thrombin or thrombin fibrinogen to fibrin. Fibrin, its network plus their damage form element of blood are trapped in it. A clot या coagulum reddish brown scum. Mechanism of blood clotting. Enzyme cascade theory proposed by McFarlane and co-workers. According to this theory, there are three steps in blood clotting: releasing of thromboplastin, injured tissue synthesized exothromboplastin, and platelets synthesized endothromboplastin. Both these thromboplastin react with plasma protein in the presence of calcium ions to form prothrombinase enzyme thrombokinase. This enzyme inactive heparin antiheparin. Conversion of prothrombin into thrombin. Prothrombinase enzyme convert inactive prothrombin into active thrombin in the presence of calcium ion. Number third, conversion of fibrinogen into fibrin. Fibrinogen is soluble protein of plasma thrombin protein polymerase monomers of fibrinogen to form insoluble fibers protein fibrin. Fibrin fibers form network on cut or wound in which blood corpuscles got trapped. This form clotting of blood. After clotting, a pale yellow liquid oozes from clot called serum, in which antibodies are formed. Blood corpuscles, blood minus corpuscles, plasma minus fibrinogen serum. Clotting factors. 13 clotting factors help in blood clotting. These factors are mainly produced in liver. Vitamin K is required in the synthesis of these clotting factors. These factors are represented in Roman number. Blood group. ABO system. As you know, blood of human beings differ in certain aspects, though it appears to be similar. Various types of grouping of blood has been done. Two such grouping as the ABO and RH are widely used all over the world. ABO grouping is based on the presence or absence of two surface antigen chemicals that can induce immune response on the RBC, namely A and B. Similarly, the plasma of different individuals contain two natural antibodies protein produced in response to antigen. The distribution of antigens and antibodies in the four group of blood A, B, AB and O are given in table. You probably know that during blood transfusion any blood cannot be used. The blood of a donor has to be carefully matched with the blood of a recipient before any blood transfusion to avoid severe problems of clumping, destruction of RBC. The donor's compatibility is also shown in the table. ABO discovered by Landsteiner, AB discovered by De Castello and Sturley. Blood group and donor capability. Blood group A, B, ABO. Blood group A, antigen A, antibodies anti-B, donor A and O. Blood group B, antigen B, antibodies in plasma anti-A, donor B and O. AB blood group. Antigen on RBC is AB, antigen in plasma no. Donors group AB, A, B, O. O. No antigens on RBC, antibodies A, donor groups B, O. From the above mentioned table, it is evident that group O blood can be donated to person with any other blood group, and hence O group individuals are called universal donors. Persons with AB group can accept blood from person with AB as well as the other group of blood. Therefore, such persons are called universal recipients. RH factor discovered by Landsteiner and Weiner in rhesus monkey. RH antigen is due to dominant gene, so if one of the gamete possesses gene of RH factor, its offspring will be RH positive. RH antigen found on the surface of RBC. Another antigen, the RH antigen, similar to one present in rhesus monkey, hence RH is also observed on the surface of RBC of majority, nearly 80% of humans. Such individuals are called RH positive. RH positive and those in whom this antigen is absent are called RH negative. Population. RH antigen, RH plus person present, RH antibody in plasma absent. Percentage ratio in India 97% RH positive and 80% ratio in world RH positive. An RH negative person RH antigen absent. Rh antibody in plasma absent. Ratio in India Rh negative percent 3% and ratio in world Rh negative percent 20%. An Rh negative person, if exposed to Rh positive blood, will form specific antibodies against the Rh antigen. Therefore, Rh group should also be matched before transfusions. A special case of Rh incompatibility mismatching has been observed between the Rh negative blood of a parent mother with Rh positive blood of the fetus. Rh antigen of the fetus do not get exposed to the Rh negative blood of the mother in the first pregnancy, as the two bloods are well separated by the placenta. However, during the delivery of the first child, there is a possibility to exposure of the maternal blood to small amounts of Rh positive blood from the fetus. In such cases, the mother starts preparing antibodies against Rh in her blood. In case of her subsequent pregnancies, the Rh antibodies from the mother Rh negative can leak into the blood of the fetus Rh positive and destroy the fetal RBC. This could be fatal to the fetus or could cause severe anemia and jaundice to the baby. This condition is called erythroblastosis fetus. This can be avoided by administering anti-Rh antibodies to the mother immediately after the delivery of the first child. Lymph. As the blood passes through the capillaries in tissue, some water, along with many small water-soluble substances, moves out into the space between the cells of tissue, leaving the larger protein and most of the formed elements in the blood vessels. This fluid released out is called the interstitial fluid or tissue fluid. It is the same mineral distribution as that in plasma. Exchange of nutrients, gases, etc. between the blood and the cells always occur through this fluid. An elaborate network of vessels called the lymphatic system collects this fluid and drains it back to the major vein. The fluid present in the lymphatic system is called the lymph. Lymph is a colorless fluid containing specialized lymphocytes which are responsible for the immune response of the body. Lymph is also an important carrier of nutrients, hormones, etc. Fats are absorbed through lymph in the lacteals present in the intestinal. In RBC, carbonic anhydrase enzyme is present which increase rate of formation and dissociation of carbonic acid by 5000 times. Fastest catalyst with zinc. 1 gram hemoglobin carries 1.34 ml O2. 100 ml blood contain 15 gram hemoglobin. 100 ml blood transport 20 ml O2. Size of RBC. Largest RBC Amphineuma 75 to 80 micro. Class Amphibia. Smallest RBC Musk deer 2.5 micro. Class Mammalia. Largest RBC among all mammals in elephant 9 to 11 micro. Newborn baby 100 days. Rabbit 80 days. Frog 100 days. Increase in the RBC count condition is called polycythemia. This condition occurs at hill station. Kidney is an erythropoietic organ in frog. In resting and slow flowing blood, the RBC forms pulse called rolox by adhering together due to surface tension. Minute bits of disintegrated red blood corpuscles is known as hemoconia. In female neutrophils, where body is attached with lobe of nucleus, which is formed by the modification of X chromosome, it helps in sex detection. AB blood group is discovered by D. Costello and Asturli. Oxygen association of hemoglobin fetal is more than adult hemoglobin.